Okay, so we're going to explore the basal ganglia in a little bit more detail. The go side, remember this is behavior activation, this is movements. And then the no-go side, this is the inaction, the suppressing of movements. So we've kind of had a couple of different videos so far. We'll just recap these spots so to make sense of them. So we have the input areas, the caudate and the putamen. And then the relay stations, kind of like relay stations, are in purple. The globus pallidus external, subthalamic nuclei are here. And then these blue areas are the outputs. Globus pallidus internal. Remember, the globus pallidus kind of makes a triangle. The external and the internal. Why this is external is this is the midline. This is right down the middle. So ipsilateral is just one side. Contralateral is both sides. Now, the way this is laid out, the go side is just on one side, and the no-go side is on the other side. That's not the way it works. This is just for illustration purposes. So we have these output areas that are projecting up to the thalamus, globus pallidus internal, and the substantia nigra pars reticulata, which are here. This is the substantia nigra. The reticulata is all GABA. The compacta is all dopamine, both excitatory, D1, and inhibitory, D2. We're going to talk about these today. We're going to talk about a cool little feature, unknown feature. I'll just tell you, it's not well understood, or the textbooks doesn't teach this. So we're going to learn about this. The globus pallidus external has a special cell that is coming into more notification. It's coming into a spotlight. And the spotlight's going to be fitting here because it projects back up to the dorsal striatum. Remember, the caudate and the putamen make up the dorsal striatum. These are the input areas. So a good way of thinking about this, because it's not like this is activated, so this is inactivated, or this is activated and it finishes, and then it goes on to the next sequence. Maybe it goes to here. This is always firing. These are just a lot of electrical projections are happening here. This is nonstop whenever we're awake, essentially. So let's talk about something that modern humans are doing too much of, quite frankly, too much of it. It's typing. Typing requires very precise movements and sequencing. It's a space-time thing. That's what we're always talking about here. It's the space-time thing because this is all timing. So you're typing a word, you're hitting a letter with precise timing. If you have inaccurate timing, you're going to flop the letters. So what you're trying to type is not going to make sense. There's very precise go, no-go sequencing here for typing, or something else is people are on the phone using their thumbs way too much. You people are robots. You people are just slouched over robots, and you can just type with such precision on the phone with your thumbs, and it's go, no-go actions, very fast, precise actions. That's all right here. Everything we do movement-wise is running through here, except for those goofy reflexes, like if you hit your elbow or you hit your kneecap with a little hammer. So this is the go, no-go area again. So let's zoom in and really talk more about the no-go area. But anytime we talk about one of these pathways, the other one is very much involved. But let's just highlight the no-go area, because I want to talk about the globus pallidus external. Remember, this is only active in the indirect no-go pathway. This is not hit in the go pathway. So the no-go pathway. It is well understood that the globus pallidus external will send projections to the subthalamic nuclei. The subthalamic nuclei will then touch the pars reticulata and the globus pallidus internal so that they can stop, kind of suppress the thalamus. Okay? Remember, this is all GABA. All of this is inhibition. We're going to talk about a special inhibitory cell. What is not well known, those cells projecting to the subthalamic nuclei and the pars reticulata are called prototypic. Well known. Those are prototypic cells. And this makes up the majority of these projections that are running through the indirect pathway. 70 to 80% of them, actually. And parvalbumin is involved with this because this is an inhibitory cell. You can find all the data you want on prototypic cells. The other cell type, right here, Arkeypallidial. Sometimes it's called Archie for short. It's understandable. So if we think about the role of this globus pallidus external with this precision that we talked about, with the timing, or maybe it's hitting a baseball, whatever, any type of movement. These have very fascinating little features because they speak back up to the dorsal striatum. This isn't well understood until maybe 10, 12 years ago. So this is really a hot spot with neuroscience, are these Arkeypallidial cells. These are very powerful, and just one projection back up to either the putamen or the caudate nucleus, one projection back up will touch on thousands of those medium spiny neurons. Remember, these have medium spiny neurons, which are a little bit different because they have the larger spines. And the dopamine pars compacta will send up those D1 and D2 like for excitatory and inhibitory action to help these dorsal striatum, these input areas, to kind of determine, do I go or do I not go? This is why this is a go, no-go balance. And remember the precision that it requires. So what are some of the functions of these types of cells from the globus pallidus external back up to the dorsal striatum? Why the need to talk back up to the dorsal striatum? Well, in large part, these two nuclei, the dorsal striatum, will determine what our actions are. Remember the example with the OCD, or anything really. We extracted a specific cell from here, and we drew it out, and we highlighted a specific spine, and that specific spine, every time there's a connection, that specific spine in that specific cell, it means the same thing. So as the caudate and the putamen are activated, it knows, based off of the cells involved and the value involved, what action that the living organism is going to carry out based off of how that living organism is constructed. Remember, right here, within the basal ganglia, but more specifically, the dorsal striatum. Who defines motivation? We don't necessarily define motivation. Our synaptic connections and who we are within our central nervous system does. This is what defines motivation, because you have to view it from the sense of being a central nervous system. You're just going to respond. That's all the central nervous system wants to do. It doesn't like to work. Remember, we talked about the habits. A lot of habits are here, especially laterally. That's because the living organism prefers habits. Why? Because it automates responses. We don't have to think about things, and it conserves energy. The brain, one of the top goals of the brain, what it's trying to accomplish is conserve biological energy. And whenever things are just habit, you don't have to think about things. You don't have to work. It'll just respond. Remember those connections that are bigger. This is called morphology. These things grow in size. These bigger connections become preferred. So the brain will just send all of its signals to there. So with this Arkey cell, it has some functional roles here. And a big role here, because caudate nucleus and the putamen is determining our actions. So these cells speaking back up to the dorsal striatum will help pause it. It has action cancellation. Okay, not only like completely, but maybe it's just a pause. Remember, we talked about the sequencing and the timing of typing. Our key cells are heavily involved in this precise movements like this. Another thing that projecting back up to this will allow is to focus and kind of reevaluate. This pause here allows those inputs from the cortex. Remember, some of these dorsal striatum inputs are from the cortex, the prefrontal cortex. In large part, you can think about these cell types are very similar to the ACC. We talked about the ACC in quite detail. They have very similar roles. These provide a large input to the dorsal striatum because of the conflict monitoring, evaluating risk reward, action selection type of things. The third role here is the sequence and timing. There is so much going on here that just like the thalamus has little assistants like the thalamic reticular nucleus and the subthalamic nucleus, these are kind of like assistants to the thalamus because it's a workhorse. These cells right here are going to be like little assistants to the caudate and the putamen. That is its kind of role here to help direct, because this is what we're doing. The dorsal striatum can be thought of as an orchestra or a conductor to the orchestra. Why? Because that's what these dopamine cells are actually doing up to here. These dopamine cells, be it D1, the excitatory, or D2, the inhibitory, is conducting this go or no go based off of these excitatory or inhibitory neurons, activation versus inactivation. So these cells will kind of help this, because whenever I said that one projection back up will touch around 1,000 medium spiny neurons, that's it. These cells right here are acting on those medium spiny neurons as well. So it's kind of like an assistant, because you need a lot of traffic control here. When things are becoming overwhelmed and so forth, these cells can speak back up and help quiet so that they can evaluate, pause, reevaluate, and then proceed. This is a big part of what these cells are doing, and it's very unknown. Why I'm bringing this up with autism, and remember the Parkinson's episode, is because there's a lot of no-go implications. If you think about stereotyping and those just awkward movements, this is a lack of control of this no-go area, because this is inactivating movements. So with things like autism and Parkinson's, there's a lot of uncontrollable movements. It's right here. It's right here with these types of cells and the prototypicals and the substantia nigra pars compacta. So uncontrollable movements will be in large part the globus pallidus, the external and the internal, because these are GABA as well. Substantia nigra pars reticulata for the GABA, because it's help suppressing the thalamus, and the substantia nigra pars compacta, dopamine, GABA. This is a pretty good example here that handwriting is very much involved with this. Every action is involved with this. So the uncontrollable movement in large part is just these no-go areas being deficient. A good way of thinking about this, and a good way of thinking about many things in neuroscience, is the seesaw. We've driven this out before. Excitatory on one side, inhibitory on the other side. It's no doubt that Autistics have fewer inhibitory cells. So what is this fulcrum? Could be a lot of different things. It could be a prefrontal cortex. It could be dopamine from the pars compacta. It could be these cells right here, speaking back up. There's a lot of different things that can influence this balance. But one thing that the Arkeypallidial cells are trying to do is balance this out appropriately. We want this in a healthy range, depending on the context. So if excitation is higher, maybe we want it higher. It just depends on the context. Maybe we're running, maybe we're trying to survive, maybe we're typing fast, and so forth, right? So this is just constantly shifting. But when we have this healthy balance, maybe to have this action cancellation and global pause, that way we can reevaluate if we are hijacked to the high excitation. This isn't happening. This isn't happening. And so this isn't happening. Remember, this is a large input to the dorsal striatum. So this is all imbalanced. One way that you can think about these special cells here is the dorsal striatum's input area for pause and spotlight system. And this will globally quiet competing neural circuits, competing actions, while sharpening a single and clean outcome. Whatever the living organism's desire is that matching this sequence? Is it an adaptive response? Remember, this is a part of the medial prefrontal cortex. The ACC and the medial prefrontal cortex, it's probably the largest role is adaptive responses. Are we adapting to the environment as we're navigating the world, the complicated social world? This is all a part of this. This is all a part of every movement. So a little bit more on the basal ganglia and the no-go side.